In this video, we'll be spending some time to look at the various proteins in the cell membrane. And they do a number of things, and we're just going to look at some general models as to what they do. So let's start off by having a look at one of the largest categories, and that would be of a receptor. And of course, receptors often deal with chemical messages that come in. Now, many times these chemical messages cannot go through the cell membrane directly and have to use a receptor on the outside to transmit the information or the message inside the cell. So receptors are a very large category. And a receptor binds a chemical messengers. You know, one example of this, but there's very many examples, would be things like hormones sent by other cells. The other type group of proteins that we'll often see in a cell membrane are enzymes, that they'll be embedded in the cell membrane and they can break things down. Now, one of the cases, once again, there's many other cases of this, is that often we'll see that enzymes are there to break down the chemical messages, because you don't want these chemical messages to linger here forever once they've acted. So the enzymes can actually break them down and basically end its effect. Another large group would be the channels. And the channels are designed for things that cannot get through the cell membrane on their own, because we're looking at issues with hydrophilic, hydrophobic incompatibility. So the channels, like a channel protein, will allow certain things of the right chemistry, right size to enter the cell and or to leave the cell. So channel proteins, they're constantly open and allow solutes to pass in and out of the cell. Now, another one that looks very similar to that, the basic channel, is a gated channel. And this is different because this one can open and close. So it opens and closes to allow solutes through. So you can see here it's in its closed state, but it can open and allow for the passage of solutes. But what this allows for is basically regulation as to when things come and go. And so it's not constantly open. The basic channel, the regular channel, is always open and allows things to come and go, but the gated channels are only open at certain times. And we'll see some examples of those as we go through the course. Another group are the cell identity markers, or cell identification markers. And you can see in this case we have a protein embedded in the cell membrane, and then coming off the top you can see this is a basically a carbohydrate chain. So this is a glycoprotein, and they act as cell identification markers. This is very important for the immune system to recognize self versus non-self. And once again, when we get into things like the immune system, we'll spend more time looking at that. Another group are the cell adhesion molecules. Often they're just referred to as CAMs, for cell adhesion molecules. And what these allow the cell to do is basically anchor itself. In this case, we can see here's one cell adhesion molecule sticking out of the plasma membrane, and here's an adjoining cell with its cell adhesion molecule. Almost think of it as like Velcro, to help Velcro these two cells together. Sometimes the cell adhesion molecules will actually Velcro to things in the matrix. Could be fibers in the matrix, etc. Now, we're going to talk a bit about special types of membrane proteins, and we talked a bit about receptors that bind to chemical messages. There's a very important system. Now, we're going to look at the general system. The detail of the system as to which enzymes get turned on or off varies from cell to cell and from moment to moment, even within that cell, and the system that you're going to use. But we're going to look at this general thing called the second messenger system. And this is a way that the environment the cell is in can communicate with that cell and help it receive chemical messages. And we're going to spend time in the next video to really look at that in detail. Also, enzymes, which we mentioned. And remember, enzymes are there to catalyze reactions, digestion of molecules, etc., and may actually have a role in in the second messenger system in terms of stopping the function. And once again, we'll look at both of those in the next video, where we're going to look at the signal, the second messenger system, and talk briefly about its deactivation. How do you turn it off? We also have channel proteins, and we mentioned those and we saw them, is that channel proteins allow hydrophilic substances and water to pass through the membrane. Because remember, the middle part of that lipid bilayer membrane is hydrophobic. So if you're hydrophilic, you'd have a hard time going through the membrane, so these channels allow you to get in and out. Now, some are open, which was what we saw in the last slide, and some are gated. Now, here's some very important terminology coming up, and that is gated channels versus voltage gated channels versus mechanically gated. And this is going to become very important in upcoming chapters, such as in the nervous system, when we talk about the functioning of neurons. And really what we have is, in a gated channel, it responds to, or I should say in a ligand gated channel, it responds to chemical messages. Now let's look at the word ligate or ligand. To ligate something is to tie together, to stitch it together. So this is a type of channel that actually needs something to combine onto it or bind onto it in order to have its activation. The next group were the voltage, and not surprisingly with a name like that, is it responds to change in charges in the local environment. So these type of gates will only become activated if the membrane close to the gate has a voltage change occurring, and then it'll open. And then mechanical gates are those that respond to physical stress on the cell. And these are really critical to nerve and muscle function. So just to remind you, the three types of gates you can have in terms of the ones that can open and close are the ligand. You need something to bind onto it. Voltage gates, where you actually need a cell membrane voltage change, or mechanical, where the gate is responding to some type of physical stress on the cell or the local area. There's also carriers, and they bind solutes and transfer them across the membrane. Now, some carriers require ATP, and if that's the case, we tend to call them pumps, like the sodium-potassium pump. And just one heads up that if you're a carrier that's going to expend ATP, it often suggests that you're moving solutes from an area of low concentration to high concentration, so they're going against their concentration gradient. And we'll see examples of those. The other thing that we saw in the diagram a couple slides back were the cell identification markers, glycoproteins that act as identification tags, and we looked at cell adhesion molecules that mechanically basically link you to the extracellular material or your neighboring cells. Okay, we're going to stop there, and on the next video we're going to come back and start talking about a very important process called the second messenger, or the second messenger system.